So someone wanted to know about what is the strategy in the clinic for determining what genetic testing to do on people who have eye disorders in the neuro-op clinic. So we're really talking about ophthalmic genetics, not the whole field of genetics. That would take too long. And the first thing we have to do is identify the phenotype. So the phenotype, what are we dealing with clinically? Because genetic disorders have specific presentations. And so that is the in vivo assessment. The in vivo assessment is in the live patient, talking to them and examining them. So we'll just use as a prototype, acute or subacute loss of vision in both eyes with a central or secocentral scotoma and a disc that looks a little bit swollen but isn't swollen or has peripapillary telangiectasias in a young male. So that is the prototype phenotype for Leber hereditary optic neuropathy. So in this kind of clinical scenario where the in vivo phenotype is so classic, we would go to targeted single gene testing. And because 95% of the mutations are just three, 11778, 14484, and 3460, there are commercial targeted gene panels that would apply to this person right here, where you don't have to do the whole genome or the whole exome, because this would cover 95% of the cases. If, however, this was negative, then 5% of the cases are not covered by that. And so we'd go to the next level up, which is the whole exome. And if you still thought it was genetic, the whole genome. So this is the intron included versus not including the intron, just the coding sequences. So normally, if the phenotype, the in vivo, is so classic, then we'll go to a targeted in vitro assessment, serum testing for a specific gene, and then you can go to whole exome and whole genome. The problem with going to whole exome and whole genome is you get all these gene results, and those variants might be of an uncertain significance, or they might be of a certain significance. So what that means is on a spectrum of pathogenicity, some things are clearly benign polymorphisms, and some things are clearly pathogenic and they cause the disease. And then you got these variants of uncertain significance. So it's probably benign or probably pathogenic. And so to answer this kind of question, we need in silico. In silico means you're going to dive into the databases, you're going to look for analogy from other countries where they have that same gene and see if it's in the other populations. You're going to try and identify the protein product and see if that protein is associated with the same phenotype. And that is what we mean by in silico. So the strategy for in the clinic ophthalmic genetic testing: in vivo assessment of the phenotype, in vitro assessment for the labs, targeted whole exome, whole genome, or panels, and if you identify a variant of uncertain significance, determining in silico whether that gene is benign or pathogenic by looking at conservation across species, other databases that show whether that gene has been reported before, and trying to move the variant of uncertain significance towards either pathogenicity or not. And that's completely dependent on the in vivo phenotype.
My question is,